Hello children, in video 5 we are going to have a close look at the first process of protein synthesis, that is transcription. I will also give you some visuals so that you will have a clear idea of the process of transcription. As already indicated, what are the requirements of transcription is the transcriptional unit and the enzyme involved in transcription, that is RNA polymerase. But first of all, what is transcription? A recap of the definition of transcription. DNA acts as a template from which an mRNA is synthesized. This process is called transcription. So, the process by which an mRNA is derived from a DNA is known as transcription. The enzyme involved is RNA polymerase. And as I told you, this enzyme functions only in the 5' 3' direction. So, which strand of the DNA works as a template for transcribing into mRNA? It is the 3', 5' strand. So, if we have the two strands, this is running the 3', 5', and this strand is running the 5', 3' strand. This strand is on which the mRNA is going to be transcribed by the RNA polymerase. Now, we have to figure out the two genes, very important genes. The start gene, which is known as the promoter, and the stop gene, which is known as the terminator. So, only this part is going to be transcribed, and that too, that segment, and that too, the strand which is running the 3', 5' strand. So, if here it is AAA, CC, GG, what will be the RNA, the mRNA that is going to be transcribed? It will be, for A it is UU, for C it is GG, and for GG it is CC. So, this is going to be the mRNA that is going to be transcribed with the help of the enzyme RNA polymerase. So, we are going to see how is this transcription occurring with the help of this enzyme. Actually, this RNA polymerase functions only for polymerization. It only helps in elongation, that is by adding the appropriate nucleotides or bases. It helps in only elongation, but to initiate and to terminate it should get associated with some factors. For example, if the RNA polymerase is represented by this figure, this will only add nucleotides. RNA polymerase alone can only add nucleotides. If suppose the nucleotides on the strand is AA, GG, CCC, it will just add the appropriate nucleotides or bases. For AAA, it's U, for this it's C, and for this it's G. It only adds the nucleotides, and this process called polymerization. For it to initiate, it should bind with a factor called sigma. And for it to terminate this process, this RNA polymerase should bind with another factor called Rho factor. So, RNA polymerase alone only adds nucleotide, only adds base pairs. For it to initiate, it should get associated with a sigma factor, and for this process to get terminated, it should get associated with another factor called the Rho factor. This is depicted in this diagram. So, the process of transcription involves three phases: initiation, elongation, and termination. In initiation, as I told you, RNA polymerase binds with a sigma factor. And then where will it bind? This combined holo enzyme. Now, this RNA polymerase plus sigma factor forms an holo enzyme. This enzyme binds at the gene called the promoter. Let this be the promoter. It binds here. It comes and binds here. When it binds here, what happens immediately? Unwinding takes place. Unwinding takes place up till the terminator. There is another gene I told you to figure out, is the terminator. So, from here to here, unwinding takes place. So, the entire coil structure will appear like this. unwind. It will unwind. So that's the first step called initiation. Second step is elongation. In elongation is just addition of the appropriate nucleotides. If suppose the nucleotides were A, C, C and G, the RNA polymerase will bring the bases which are complementary to A, that is U, then for C, G, and for G, C. So that's how this yellow strand comes out. Okay, and this continues till where? Till it reaches the terminator. So this is called elongation. Elongation is just simply adding of bases or polymerization by RNA polymerase. That is done alone by RNA polymerase. See, is it associated with any factor? No. Then last is termination. Now you can see this RNA polymerase binds with another factor called the Rho factor for this to terminate. And what happened? Everything is released. The mRNA is released. The coil of the DNA binds back and the RNA polymerase detaches. So the three steps are initiation, elongation and termination. RNA polymerase alone does the function of adding nucleotides, adding bases complementary to the strand which is running in the 3', 5' direction. For it to initiate, it should get associated with which factor? Sigma factor. And for it to get terminated, it should get associated with a factor called the Rho factor. That you should remember. So therefore, we can divide these into three steps: initiation, elongation and termination. In initiation, what happens? RNA polymerase binds with a sigma factor and this forms a hollow enzyme which binds to the promoter. And once it binds to the promoter, what happens? Unwinding occurs. Second step, elongation. Elongation, what happens here is the sigma factor detaches. The RNA polymerase alone adds on nucleotides. And this continues till the terminator is reached. Once the terminator is reached, what happens is the RNA polymerase attaches or associates with the Rho factor. So termination is brought about. When termination occurs, what happens? Everything detaches. That is, the mRNA that is formed detaches, the RNA polymerase detaches from the DNA and the DNA coils back. So let's see this with the help of a small video clipping. Here you can see this is the RNA polymerase. It binds to the promoter on the DNA. And there are three stages: initiation, elongation and termination. First, initiation. See, this is a promoter which is highlighted. Now the RNA polymerase binds. When the RNA polymerase binds, what happens? Slowly you notice the nucleotides are going to come, appropriate nucleotides are going to come. Before that, unwinding takes place. The first step is unwinding. See, the two coils unwinds and look, the nucleotides are added and look at the direction. It adds in the 5', 3' direction and look at the complementary base pairing. And a new strand is formed, and this new strand that is formed is called the messenger RNA. This continues till the termination region is reached or the terminator gene is reached. Once it reaches the terminator, another factor called Rho factor binds and all the structures gets released. Now we get the mRNA out of the DNA. For eukaryotes, this mRNA is not final. It contains certain regions called introns. They are the junks that do not code for proteins, and other region called exons. It should therefore undergo a process called processing, where at the tail end an adenylate residue is added and at the 5' end methyl guanosine triphosphate is added, and the introns are removed with the help of spliceosomes. See, all the introns are removed and exons are joined. Look, now we have the final mRNA. This is the final mRNA which comes out through the nuclear membrane into the cytoplasm, ready for the next step called translation. Now you see in translation, the mRNA is ready with all the bases which are in the form of codons. Look at the codons. 3 consecutive bases forms the codons, and you have to make a reference with the genetic code to find out which amino acid comes to the site. This is called the translational complex. In the video we saw eukaryotes. In eukaryotes, the mRNA that is synthesized is not final. See, this mRNA that came out is not final. It has to undergo some processing. The processing, there are 3 steps. One step is capping, next is tailing, and very important is splicing. Capping is done at the 5' end, tailing is done at the 3' end. Capping is done with the help of methyl guanosine triphosphate is added at the 5' end, and tailing is done at the 3' end with the help of an adenylate residue. And splicing, what is splicing? The introns are removed. You can see in this picture. As I told you, this is not final. This mRNA formed is not final, and hence this mRNA is not called mRNA, it is called a heterogeneous RNA, hnRNA. It has both introns and exons. Introns are junks. They are not required. They do not code for protein. So, the introns should be removed. So, that is done by the help of splicing. See, this is the introns. If I am going to highlight in blue, introns, interrupting. There are some introns, and yellow are the exons. What is required is the exons. So, what is done? The introns are removed. The process of removing the introns is called splicing, and that is done with the help of a scissors called spliceosomes. And finally, you see, you got only the yellow strand. See, yellow. Only all the exons are joined, and finally you have to cap and tail it. Capping is done with at the 5' end with methyl, it's easy to remember, methyl guanosine triphosphate, and tailing is done at the 3' end with an adenylate residue. Okay, now this is the final mRNA. So, here I can highlight another difference between prokaryotes and eukaryotes. In prokaryotes, the mRNA that is formed as a result of transcription is final. It does not require processing. Whereas, in eukaryotes, the mRNA that is formed is not final. It is called hnRNA. It has to undergo a process of processing. It requires processing, which involves capping, tailing and splicing. Splicing is the removal of the introns. Introns are the regions that are junks. They do not code for protein. So, they have to be removed, and all the exons should be joined. And finally, we have to cap at the 5' end with methyl guanosine triphosphate and tail it at the 3' end with adenylate residue. And now we have a final mRNA. So, that is going to be the 4 difference between the prokaryotes and eukaryotes. So, the 4 differences: prokaryotes, the mRNA formed is final, whereas in eukaryotes the mRNA formed is not final. It is called a hnRNA. It requires processing, and processing occurring in 3 stages: capping, tailing and splicing. Now, in the next video we are going to see the next phase called translation.